In this short video, we will be discussing some basic concepts of cell biology. I am Sufyan Akram. Now, to understand the structure and functions of the human body and how it responds to challenges and diseases, it is important to understand the anatomy and physiology of the cell. In a multicellular organism like ourselves, cells specialize to perform their required functions. For example, muscle cells contract, nerve cells transmit impulses, and skin cells protect. Purpose of this short video is to explore those basic functions of a cell. Now, this lecture is just an overview. For detailed discussion of the topic and to study some clinical examples, please consult the textbook. So in this first part, we will be looking at cell structure and functions and cell division. Now, each cell has a structure that is almost as complex as the human body. For example, each cell contains as many molecules as the body has cells. And the body on average has more than 1 trillion cells. Each cell is surrounded by a membrane called plasma membrane or cell membrane. Within the cell, there is a structure known as nucleus, and this contains DNA or chromatin. There is this thing called cytoplasm, which is a fluid that contains a lot of organelles and dissolved substances. What we will do now, we will look at these components one by one. So let's focus first on the cell membrane or the plasma membrane. Basically acts as a barrier separating inside and outside of the cell. It controls the flow of substances into and out of the cell, and it acts as a selective membrane. It allows only certain substances to pass through. It helps identify the cell to other cells. The structure of the membrane is lipid bilayer, and the main lipid is phospholipid. As you see in this picture, the one in light blue color. But within the lipid bilayer, you see a lot of other structures floating. These are proteins and glycoproteins, and they have a lot of functions. In the next slide, I will try to capture some of the functions of these dissolved proteins and glycoproteins. These could function as ion channels, forming a pore through which different ions can pass. They can act as carriers, so they transport a specific substance across the membrane, act like gates that could close or open, and they allow only a specific substance to pass through. They could act as receptors, identifying certain chemicals or ligands and then responding accordingly. They could act as enzymes. They could also act as linkers or anchors, allow cells to adhere to each other or to the basement membrane. They could also act as cell identity markers. For example, it allows us to distinguish our cells from other cells or foreign cells. Now, transport across the plasma membrane occurs by two mechanisms: passive or active. Passive transport does not require energy, and there are a few types. Number one is simple diffusion, which is movement of solutes from an area of high concentration to low concentration or down the concentration gradient. The second type is facilitated diffusion, which is again similar to simple diffusion. It is down the concentration gradient, but it is facilitated by integral proteins. As we saw in the previous slide, there are certain proteins which act like channels, so molecules can slip through those channels or pores. Third type is osmosis. Now, osmosis is not the movement of solutes, it is rather the movement of water or solvent. Water travels through a selectively permeable membrane so that concentrations of the solute are same on both sides of the membrane. For example, if you have a solute that cannot penetrate through the membrane, the water will move across the membrane to make sure that the concentration of this solute is equal on both sides. And osmosis is vital for the normal fluid balance in our body. And when we study fluid balance and certain diseases, this will become more clear. Filtration is similar to osmosis, except that pressure pushes water and solutes against the cell membrane. Let's look at osmosis more closely now. Imagine a container where you have water and there is a selectively permeable or semi-permeable membrane in the middle. So this is cell membrane or plasma membrane. And on one side, you have certain molecules or solute which cannot pass through the membrane. Okay? On one side, there is a high concentration of this solute. On the other side, there is a low concentration, or there is no solute there. Okay. What will happen now? This membrane allows water to pass through. Which way the water will move? The water will start moving to the side where you have higher solute concentration. Okay. Let's look at the active transport mechanisms now. As I told you earlier, the active transport mechanisms require energy. And there are a few types. There are active transport carriers or pumps. They can transport molecules against the concentration gradient. Remember, in passive transport, the molecules can only move down the concentration gradient. In this one, you're moving the molecules against their concentration. That's why you require energy. An example is sodium-potassium pump, endocytosis and exocytosis. So endocytosis is where you move the molecules inside the cell. This could be via pinocytosis or phagocytosis, depending upon the size of the molecules you want to move across. Exocytosis is where you're moving the molecules out of the cell. And these processes will require energy, as I told you. Cytoplasm is a ground substance, as I told you earlier, also known as matrix, in which various cellular components are found. It is 75 to 90% water, but there are solid components, mainly carbohydrates, lipids, and inorganic substances. And it is the substance in which all chemical reactions occur. Let's look at the nucleus now. When considering the nucleus, a simple analogy is to think of it as the brain of the cell, because it contains what is known as DNA, which is the genetic material. Cells constantly face challenges. They age and they die. Remaining cells then divide to replace the cells which are lost. This happens through a process called meiosis, where a cell passes through an ordered series of events in which the cell duplicates its contents and then divides into two cells. This process of cell division must be highly ordered and tightly regulated. Meiosis, on the other hand, is the type of cell division which occurs in sexual organs like testes and ovaries and leads to formation of gametes. Mitosis can be divided into four stages: prophase, metaphase, anaphase, and telophase. Now, cells before and after these mitotic phases remain in a stage called interphase. During these subphases, extra organelles are manufactured, DNA undergoes replication, the cell builds up a store of energy that is required for the process of division. So everything is duplicated before the cell will commit to mitosis. Now let's look at these phases a little more in depth. During prophase, the chromosomal material condenses to form compact mitotic chromosomes. The cytoskeleton is dismantled and mitotic spindle is assembled, as you see on the left side of this diagram over here. This is called mitotic spindle. Nuclear envelope or the nuclear membrane also disappears. So chromosomes are now in the cytoplasm. In prometaphase, the chromosomal microtubules attach to the kinetochores of chromosomes. So at the middle of the chromosome, during metaphase, you see the chromosomes are nicely aligned in the middle. During anaphase, these chromosomes split. So one copy moves to one pole and the other copy moves to the other pole. Further down, the last phase, which is called telophase, you see one set of chromosomes that move to one pole, the other set to the other pole. After this, what happens is cytokinesis, or the cell divides into two, each daughter cell receiving one set of the chromosomes. If you look at the bigger picture now, and if you imagine the whole cell cycle as 24 hours, then the process of replication or mitosis would last only about 1 hour. The rest of the time, the cell is undertaking the replication of DNA. It also has to produce 2 of everything that is in the cell. In addition, it also has to go through the process of obtaining and digesting nutrients so that it has the raw materials and the energy to go through these phases of cell division. So as I told you earlier, in addition to the mitotic phase, right, you have what is known as the interphase, which you divide into G1, S, and G2. As I told you earlier, this whole process has to be tightly regulated. There are many checkpoints in the cell cycle. For example, there's a checkpoint known as G1 checkpoint. There are proteins and enzymes which will make sure that the cell is big enough, the environment is favorable, nutrients are available before it commits to go into the G1 phase. After G1 phase, there is the S phase, right, when DNA gets replicated. Now, before the cell moves from S phase to G2 phase, there is a G2 checkpoint. This is where the proteins and enzymes will make sure that all DNA is replicated, and it is replicated without any errors. There's nothing missing. Environment is favorable before it goes into the G2 phase. And then there's another checkpoint which is called the metaphase checkpoint. Metaphase checkpoint makes sure that all chromosomes are aligned on the spindle and they are divided into 2 sets, and each set is moving to the separate pole of the cell. Now, these checkpoints are very important because if there's any error that occurs, these checkpoints will stop the cell cycle. The repair machinery will come into action and try to repair the damage before cell commits to the next stage. All right, let's now talk about meiosis. Each human cell has 23 pairs of chromosomes, of which 22 are autosomes and 1 set of sex chromosomes. In sexual reproduction, when ovum is penetrated by a sperm, DNA copy from both parents is combined. To ensure that the resulting zygote has the correct amount of DNA, the sperm and eggs undergo a process known as meiosis to ensure that they only contain 1 copy of chromosomes, or in other terms, are haploid. Meiosis can be divided into 8 stages, not the 4 as we saw in mitosis. Thus, there are 2 cycles of cell division. And let me show you a comparison of mitosis and meiosis. You see, in mitosis, the parent cell undergoes DNA replication, and then you have 2 daughter cells. Each one of them receives an exact copy of the DNA as of the parent cell. So mitosis generates 2 genetically identical diploid daughter cells. In case of meiosis, DNA replication occurs similar to how it happens in mitosis, and then you form 2 daughter cells. Then these 2 daughter cells divide again to form a total of 4 daughter cells. But if you compare the parent cell with these final daughter cells now, you'll see that these 4 daughter cells are haploid because they contain half the amount of DNA as compared to the parent cell. And none of these cells are genetically identical. Another very interesting point here. Why are they not genetically identical? Because during meiosis, there's something called crossover occurs. So chromosomes exchange parts with each other. These recombination of genes and crossing over results in genetic diversity. With this, we have come to the end of the first part. Thank you so much for listening, and I'll see you next time. Bye till then.